Hello everyone, welcome to my video about sarcomere structure and function. My name is Dr. Leili Hatami. In this video, I will explain what sarcomeres are and why they are important for muscle contraction. First of all, let's look how skeletal muscle is organized from the largest part to the smallest part. The largest unit is the whole muscle. This is what we can see on our body, like the biceps or quadriceps or hamstring muscle. Each of these muscle considered as an organ. So if you cut the muscle as an organ, you will see inside each muscle there are group of fibers called fascicles. So as you can see in this picture, each muscle as an organ is composed of many muscle fascicles. And then each muscle fascicle contains many muscle fibers or muscle cells, which they contract and help us move. Inside each muscle fiber or muscle cell, there are even smaller structures called myofibrils. These are long thread-like structures that run the length of the muscle fiber. Myofibrils are made up of repeating units called sarcomeres. Sarcomeres are the smallest functional unit of muscle contraction. So from largest to smallest, we have muscle as an organ, then muscle fascicle, next we have muscle fiber or muscle cell. Then we have myofibril, then sarcomere. Okay, now let's talk about sarcomeres. Sarcomeres are the basic contractile units of the striated muscle tissue. They are found in the striated muscle, like skeletal muscle and then the cardiac muscles. Sarcomeres help our muscles contract, which allows us to move. Now I will describe the structure of a sarcomere. A sarcomere is made up of thick filament and thin filament, or we can call them as the myofilament. Myofilament, thick and thin myofilament, or thick and thin filaments. Then there are Z lines made up of proteins which marks the end of each sarcomere. So you can see the Z disc or Z line at the both sides of the sarcomere. So it means the area between two Z line or two Z disc is called the sarcomere. And as you can see, inside of the sarcomere, we have the thick filament or myosin filament and thin filament or actin filament. Next, we look at the thick filaments. Thick filaments are mainly made up of myosin molecules. That's the reason we can call them as myosin filament or thick filament. Myosin has heads, as you can see in this picture. It's showing a myosin molecule, which each myosin molecule has heads, which on their head we have two binding sites, one for ATP, which means that during the muscle contraction we need the energy, so there is a binding site for ATP on the heads of the myosin molecule, and also the other binding sites are for actin. So it means that the myosin head should be connected with actin during the muscle contraction. So these heads are very important. And then you can see on the other side, we have the tails of the myosin molecule. So then many of these myosin molecule, all the head on one side and all the tail on the other side, together gonna make a thick filament. Okay, next let's look at the thin filament. Thin filament are mostly made up of actin, but they also have two proteins called troponin and tropomyosin. The actin comes together to form the long strands. These strands have binding site for myosin heads, which are important for contraction. So if you look at a myosin molecule, which is a globular molecule, that's the reason we call this as G-actin. So each of these actin molecule or G-actin, they have a binding site for myosin molecule. So remember that. On the actin, we have the binding site for myosin molecule, and on the myosin head, we have the binding site for actin, which is showing during the muscle contraction, the myosin should bind to the actin, which is going to make a cross bridge, or this connection between the myosin head and actin is called the cross bridge. Okay, let's continue. We need two strands of actin filament. So it means that we need some G-actin or actin molecules that together they're gonna make a polypeptide as an actin filament, and then we need two of these actin filament twisted together to form a thin filament. And then this tropomyosin and troponin also are important molecule, as tropomyosin gonna be attached to the actin. So you can see this strand shaped protein in this picture. It's like a brown color. It's called tropomyosin. So as you can see here, tropomyosin job is to cover the active site on actin molecule, which means that when muscle is not contracting and it's relaxed, the myosin head cannot be bind to the actin molecule, to this binding site of actin. So tropomyosin job is to block the active sites on actin molecule. So then during the muscle relaxation, myosin is not going to be attached to the actin molecule. Then the troponin molecule is also important. It has three subunits, which you can see in this picture by the yellow color. One of these subunits is going to be attached to the actin. Another subunit is going to be attached to the tropomyosin, and another one going to be attached to the calcium ion, which calcium ion is an important ion we need during the muscle contraction, which means that without calcium, we're not able to contract our muscle. So you can see that here, if there is calcium inside of the cell, inside of the cytoplasm, it's going to be attached to the troponin molecule. Okay. So then let's go and look at the sarcomere under microscope. If you look at the sarcomere under microscope, you can see some light region, dark region, light region, dark region, light region, dark region. That's the reason we call the skeletal muscle and cardiac muscle as the striated muscle tissue. You can see this striated appearance for skeletal and cardiac muscle under microscope. That is because of the placement of the actin and myosin filament in the sarcomere. So this is a microscopic picture of a sarcomere. Okay. So then you will see some dark line, which we have the z-disc. As you can see, this dark line, another z-disc, which means that between two z-discs we have the sarcomere. And then at both sides of the z-disc, you can see this light area, which this light area is called the I band. So we have the I band or light region, at both sides of the Z disk. Okay, and then in the middle of this sarcomere, we have this dark region. This dark region is called A band. So as you can see, in the middle of the A band, there is a region which is lighter than the other part of the A band. We call this region as H zone. So from here till here, which is a little bit lighter than the other part of the A band, we call this as H zone. So the H zone going to be found in the middle of the A band. And then in the center of the A band, we have the M line or M band. So make sure you know the I band, A band, H zone, and M line. This is going to be found in the sarcomere under microscope. I band means the lighter region, lighter. The A band is the darker region, and H zone is the light region in the middle of the A band. And M line is a dark line in the middle of the A band or a sarcomere. But why? Why we have this light and dark area in the sarcomere structure? We should know about the placement of the myofilaments in the sarcomere. Look at this picture. This dark blue line is showing the Z line or Z disc. And then thin filaments by this red color, you can see it's going to be attached to the Z disc. So the thin filament or actin filament are here. Let me just label this as thin filament or actin filament. It's going to be connected directly to the Z line. And then in the middle of the sarcomere, between the actin filament, we have the thick filament, as you can see by the blue color here. And the blue color is showing our thick filament. So if you can just draw it with this slide so you can learn it better. So let's draw the Z line at first, and then draw the actin filament, which they are connected to the Z line directly. So we have the actin filament or thin filament. Then between the actin filament in the middle of the sarcomere, we have this myosin filament that you can see the myosin heads on this filament. Okay, so look at here. The area which only and only and only gonna be found thick filament is called I band. So I band, which is our lighter region, it's composed of only actin filament, which they are thin filament. Okay, and then in the middle of the sarcomere, which we have the length of the myosin filament, and we see this region as dark region, is called A band. So A band would be the length of myosin filament. Okay, so if you look at the A band, you can see in some area we have overlap between the actin and myosin filament. So here we have overlap between actin and myosin. Here we have overlap between actin and myosin. That's the reason this region is a little bit darker than this region, which only gonna be found the thick filament, which is called H zone. So in H zone, we only have myosin filament. Okay, and then in the middle, when these two myosin filament are going to be connected to each other, we have a dark line. It's called M band. So if I go to this slide, you will see here the I band, which we have the lighter region, is composed of the actin filament. So we have actin filament here, only actin filament. And then from here till here, we'll have the length of the myosin filament. But in this area, we have actually overlap between actin and myosin. So if I want to draw the actin, I have to do something like that. So here we have overlap between actin and myosin. That's the reason this region is darker, but in the center of the A band or sarcomere, which we have only myosin filament, so that's going to be a little lighter than other part of the A band. So I hope you could understand the sarcomere structure under microscope. So, but again, if you can draw these for yourself, so you can understand better and easier. Okay, let's see what's gonna happen during the skeletal muscle contraction, or what are the changes in striations as the skeletal muscle contract. So if you look at this picture, it's showing this fully relaxed sarcomere. So then I'm going to label this line as the Z line and another Z line. Then from here till here would be our sarcomere. Is a fully relaxed sarcomere of a muscle fiber. And then for this picture, which is a fully contracted sarcomere, then that would be our Z line and Z line. If you measure the sarcomere of fully contracted muscle fiber and compare it with the fully relaxed sarcomere, you will see that sarcomere length has been shortened during the muscle contraction. Okay, so what can happen inside of the sarcomere? Actually, inside of the sarcomere, we have the sliding of actin filament over the myosin filament. In this sliding filament theory, it says muscle contraction happens when thin filaments slide past thick filaments. When this happens, the sarcomeres become shorter, which makes the muscle contract. And then because the thin filament slide past the thick filament, so then you will see we have more like overlap between actin and myosin. So then there is a less portion which we have the only actin filament, which means our I band shortens. Look at here, when we have only actin filament here, but then compare when now we have more overlap between actin and filament, actin and myosin. So it means that our I band shortens because of the sliding of thin filament over the thick filament, but because the length of our myosin filament not changing, the A band would be fixed. So there is no change of our A band. And then the H zone would be disappear. Because we define the H zone is the area we only have the myosin filament. But after contraction or during the contraction, we have overlap between actin and myosin. So the area that only going to be myosin filament, actually it's not there. So then you can see the H zone disappeared. So that is the changes of the striations or sarcomere during the muscle contraction. First of all, we know sarcomere shortens. So the length of sarcomere shortens. And then I band, which is only actin filament, also shortens. And H zone may disappear, as now we do not have only myosin filament. Mostly we have overlap between actin and myosin. But A band does not change. Why? Because A band is the length of the myosin molecule, myosin filament. There is no changes of the length of the any filaments. Thin filament, thick filament, length not gonna change. The only thing that we have is a sliding of the thick filament over thin filament over thick filament, then because the A band showing the length of the myosin filament, so it does not gonna change. Okay, thank you so much for watching to this video. If you have questions or thoughts about sarcomeres, please write them in the comments. I would love to hear from you guys. Thank you so much.